Hello everyone, welcome to the latest video in Omixon's Learn Me video series. My name is Adil Yuhosh and I'm going to take you through this training video today. In this video, I am going to introduce you to HLA Twin software that is used for HLA genotyping using NGS data processed by Omixon HLA typing kits. Before we start, let me show you how you can set captions on this video. To have English subtitles, click on the CC icon on the bottom right of the player. Do you need another language? No problem. Click on the settings icon, go to Subtitles CC, click on Auto Translate and select your language. Are you ready? Let's start. To begin, click on the HLA Twin icon to launch the software. You'll be asked to log in. If you are using a server version, at the first launch you will have to select which server you would like to connect to. Just double check or insert the IP address of the server and click on Connect. Each user has his own private login credentials. There are two levels of users. One, the super user, who has all permissions to change the software settings or manage other users' profiles. And two, the analyst, who has more restricted permissions used by lab technicians, for example. After logging in, you can see the main software dashboard called the Genotyping dashboard. This is the home screen from which you can start the data analysis. But let's have a quick overview of the screen first. Going from the top to the bottom, you can see which active user is logged in at any time. Also, you can find the Log Off and Close button at the top right of the screen. At the upper left corner, you can find the navigation arrows. These arrows help you to switch between different dashboards and pages of the software. In case you get lost, you can always click on the Home button and you'll be redirected to the main dashboard where we are now. Some practical information can be found next to the navigation arrows. This field contains information such as the assay version, which is selected for the data analysis, and the IMGT database version that is selected at any given time. Let's talk about the most important part of this screen, and that is the toolbar, where you find buttons for launching the analysis, for displaying the results, and for filtering of the displayed content. If you need to manage the IMGT database versions or the users or change several settings, just click on the Application Settings button. In the middle of the screen, you can see the content of your output folder with the FASTQ files. The FASTQ files are represented by the double-stranded DNA icon. Before the analysis of the files starts, there are no result files displayed, but after the analysis, the result files will be displayed under each corresponding FASTQ file. For our purposes, we have already analyzed the FASTQ files and hidden the result files. We can display them by clicking on the Analysis Filter button. The result files appear and they are represented by the table icon. A last but not least important and practical feature is the info pane in the bottom of the screen. On this pane, you can see details such as the IMGT database or software version that was used for the analysis, and so on. Let's start the adventure of NGS genotyping. In order to start an analysis, select the sample file by clicking on the file of interest, or you may select multiple files by holding on either the Ctrl or Shift keys and selecting the files of interest using your mouse. Then click on the simple genotyping button. You can also click on the Analyze button next to the sample file name to start the analysis of an individual sample. You can see the progress of the analysis on the top right of the screen in the Type a Scheduler and Event Log windows. Let's check the analysis that is in progress. Click on the Type a Scheduler button to access the running analysis tasks. In this screen, you have the opportunity to stop, remove the analysis of any file, or to rearrange the analysis file's order. Once the analysis is done, the result files are displayed under the sample files. Each result file is accompanied by the traffic lights indicating how many loci have passing QC metrics, green light, QC metrics for investigation, yellow light, and failed QC metrics, red light. Let's check the results now. You have two options for displaying individual results. You can either view the high-level results by clicking on the Results button on the right-hand side of the result file, or 2, you can view the detailed results and QC metrics by clicking on the Details button located in the same position. You can also select multiple files to display the high-level results or the detailed results on the View Results and View Details buttons respectively at the top bar. Now let's select a few result files and click on the View Result button. You'll be taken to the screen where you can check the allele calls. Here you can see each sample in a separate line. At the front of the line is the sample ID with a grey check mark indicating that the sample hasn't been assigned yet. The genotyping results are either displayed in black representing a heterozygous result, or in blue meaning it's a homozygous result. Above each result there is a traffic light. This is the quality control metric indicator and summarizes all of the QC metrics that are calculated for the locus. If you hover the mouse over it, a table pops up in the tooltip that gives you information on the QC metrics values. The chromosome symbol next to the traffic light shows the zygosity of the locus. It is another way to show whether the result is heterozygous, homozygous, or hemizygous. The next symbol you see in the row is representing the serological equivalence. Let's talk about the toolbar at the top now, where there are several functions you can use. We can start with the Setup Loci button. You can use this button to hide or display loci. By default, only the best matching alleles are displayed. This is displayed with the blue circle at each allele. These results contain 0 or the lowest number of mismatches between the consensus sequence and the reference sequence. You can toggle this filter on and off by clicking on the Best Matches Only button. The default allele precision that is displayed is the full resolution of 4 fields. However, you have the option to toggle between 2, 3 or 4 fields, depending on the precision you prefer. As you can see, the list of alleles of each sample is quite long and prevents comparing the results of multiple samples easily. In order to facilitate the result comparison, you can shorten the list by clicking on the Expand Collapse button. You will see the first allele pair in the sample line. If there are more allele pairs in the result, the corresponding part of the result will turn red, indicating at which allele level is the ambiguity. You have the option to assign one allele pair to export and report out after review. This is particularly useful when there are multiple allele pairs available at a resolution higher than the one you report, for example at 4th field resolution where you report 2nd field. To assign an allele or allele pair, just click on the light blue circle with the white check mark in it. Every allele has its own blue circle. But let me show you how to do a bulk assignment of all loci using the Assignment button. A window pops up where you can select the results you'd like to assign based on their QC metrics. Every result which is green, passed, or green-yellow, info, can be confidentially assigned. You can select other options for allele assignment as well. For your information, you can hover the mouse on the question mark to see the explanation of each option. Now you can see how many alleles have been assigned automatically. You can see that loci with green or yellow-green QC changed the blue circle into a green check mark, indicating that the result has been assigned. But there are still some loci that haven't been assigned and require further review. Let's investigate those. Select the sample and click on the Sample Details button. In the genotyping sample result page, you can see the sample results in a collapsed mode at the top table. Underneath, on the Genotype tab, the allele pairs are displayed with more details and listed locus by locus. The next tab, Quality Control, is very important if you need to check the quality of the results. There are 24 different QC metrics that are calculated for each locus separately, and they are categorized to 3 different groups: primary, secondary, and warnings for troubleshooting. Primary QC metrics are the most important ones because they describe the basic quality of your data, like number of reads, background noise, locus coverage, or allelic imbalance on key exons. The last tab, Data Statistics, contains a lot more information which may be used during a thorough investigation of failed QC metrics. You will not need to enter this tab often, but it is good to know what can be found here. First, you can see the table summarizing the read usage throughout the steps of the genotyping process. Mappability is another practical tool showing you how many reads were found for each locus and how many of them were used for constructing the consensus sequence. Scrolling down on this page, you can see cross-mapping table, allele imbalance graph, fragment size and read quality graphs. Let's now have a look at some more practical things. Let's talk about the toolbar on this screen. The first 3 buttons from the left-hand side are for displaying the gene sequence in the gene browser. You can browse both alleles together by clicking on the Browse Alignment button, or you can browse each allele separately by Browse Allele 1 or 2 buttons. The next button in the row is the Genotype Details button. In this window you can see details such as mismatch statistics broken by region, alignment statistics for average depth of coverage, and allele frequencies. The other 2 buttons, Show Mismatches and Show Novelties, help you to quickly navigate to the position of a mismatch or novel nucleotide in your gene sequence. We are now brought on the gene browser page. The gene browser is a tool primarily designed for in-depth investigation of challenging samples. You can navigate to it from the genotyping analysis results screen or from the genotyping sample results screen by clicking on the Browse Alignment or Browse Allele buttons. If you don't have any loci selected, the first result pair for HLA-A will be displayed. The fastest way to display something specific is to select the locus on the sample results screen and then use the chromosome-specific display buttons. Here you can see 4 major items of information which contain multiple tracks. The top 2 tracks contain the consensus sequence that was constructed for each allele in pair, while the 2 bottom ones contain the realignment of the short reads on the reference sequence from the IMGT database. Above the tracks you will see the nucleotide position numbering. The exact position of the cursor is always displayed in the bottom left-hand side within the metadata panel. Here you can see both the nucleotide position relative to the start of the consensus sequence generated from the sample data, as well as the corresponding nucleotide position in the IMGT HLA database. Below the position track you can see the phasing annotation. If there are no phase breaks, the whole ribbon states phased region. However, if there are any phase breaks, the ribbon will be broken at certain points and each region will be numbered consecutively: phased region 1, phased region 2, etc. In order to resolve any phasing issues, you may use the variant relationship track just below the phasing annotation. This provides information on the number of fragments supporting the relationship of the 2 neighboring heterozygous positions. The values after the word straight show the number of fragments supporting the displayed phasing for chromosome 1 and 2. The values after the word cross show the number of reads supporting the alternative cross-direction phasing for chromosome 1 and 2. Immediately below the variant relationship track comes the first consensus sequence. Both consensus sequences can be exported by clicking on the Export Sequences button. You have the option of exporting the gene browser tracks or a novel sequence. The The export of the novel sequence was designed to provide all necessary files compatible with allele submission to the gene bank and IMGT databases. If you select Export Novel Sequence, you can also export the feature table file containing a description of the gene structure. Just fill in the requested details and click on Finish button. Coming back to the sequence view, we don't see the read pileups by default, but a coverage depth graph. To view the individual short reads, click on the Visualization Mode button on the toolbar once, and then the Collapsed Mode button. You can zoom in with the mouse wheel to visualize the individual nucleotides. The two tracks on the bottom half of the screen look similar, but they reflect the reference sequence in the IMGT HLA database. The metadata panel below the sequences contains multiple bits of important information and functions. You can see the coverage depth of a certain position on a selected read, and you have the ability to copy it as well if you wish to do so. To demonstrate, let's have a look at the big picture again. Keep in mind that the first consensus track pairs up with the third track, and the second consensus track pairs up with the fourth track. If you want to reduce the amount of information displayed and compare each consensus sequence with its corresponding realignment sequence, you can use the Displayed Allele button, which allows you to switch between chromosomes to visualize one at a time. By right-clicking in the gene browser, a context menu is displayed with a number of options. Near the top of the context menu is the Show Base Statistics function, which allows you to check the exact number of reads that support the selected nucleotide position. If you would like to look for a specific nucleotide sequence within your sample, you can use the Find Sequence option to locate every occurrence of the specific pattern of nucleotides. To display only the heterozygous positions of the displayed alleles, you can use the Toggle Reference Masked option. This is particularly useful in reviewing the data. All nucleotide positions that are homozygous are grayed out, leaving only the heterozygous positions in color to highlight the differences between the displayed alleles in the gene browser. In the single chromosome view, this feature allows you to see any differences between the consensus sequence of your sample and the reference sequence in the IMGT database. In the dual chromosome view, you will also see all the positions at which the alleles in the sample are different from one another. To compare additional allele candidates in the gene browser, use the Add Custom Allele function in the context menu to add any allele from the IMGT HLA database to the gene browser alongside what is already displayed. Depending on the situation you're investigating, you may wish to investigate possible ambiguities by adding similar allele candidates. You can easily add the result of the other allele pair reported in the software by selecting Add Allele of Other Result Pair. These manually added alleles can be used instead of the actual results by using the Convert Custom Allele Pair to Result Genotype function in the context menu. These results are easily identified by the blue squared symbol in order to differentiate them from the results generated from the software, but they can be assigned, approved, and exported to reports just like the software-generated ones. Let's also assign the results that have a yellow QC indicator. Click again on the Assignment button and select Inspect QC Threshold Level. You can check the assignment of novel alleles too in order to get our novel allele assigned. At this stage, the sample is ready to be sent for approval. Select the sample to be approved, then click on the Send for Approval button. You can see that the grey check mark has now become orange and the green check marks next to the allele are striped through. From this point on, you can't change the allele assignment unless you cancel the approval by clicking on the Reject Revoke Approval button. To finish the approval process, click on the Approve Result button. You can see that the orange check mark turned green. You are now ready to export the results and generate PDF reports from the Genotyping dashboard home screen by clicking on the Export Results button. I hope you found this video useful. If you have any questions, please contact us at support@omixon.com or place an inquiry with our sales team at sales@omixon.com. For more videos, please see the Omixon YouTube channel. See you next time!